And I have any idea thank you come. Mine is going to be proper. But as some of you are aware, Microsoft let me down. And it disappeared, my latest, my most recent, therefore, those of you who may be here this evening, who are most recent members, it disappeared your email addresses. If you have received an email from me, then if you would reply, that would be good. But as you're present in the room, better than that, if you haven't replied to me, just write down your email address on my list before you go home. And that way, because I'm starting new contact lists, and I just want to make sure, because there is a lot of dead wood in the old ones. And I'd quite like to remove it. I'm very keen on deleting things. Thank you very much.
Okay, thank you, Jeremy. Good evening, everyone. I'm standing in for Professor Steve Rowland tonight. I'm Mike Moore, Vice Chairman for my students for this. I just got a little bit of domestic stuff. In case of fire, flood, plagues of frogs, or biblical events, you're not supposed to panic. You're supposed to leave by one of these two doors, please, and assemble outside. Hopefully that will not occur, but that's it. Now, before we continue, I'm just going to mention our sponsors who have been very generous to us in the past. Lansing Rotary Club provide quite a lot of money over the years, and that helps with the student event that we hold in July. And Lansing or Launceston Town Council, they have also been very generous to the society. And then just a few days ago, we received £1,000 from a chemical analytical company, DRP Analytical, partly through our chairman, Professor Steve Rowland. So that has helped our funding, and that goes towards the cost of bringing speakers from further afield. So just before we continue the talk, I'll mention next month's talk, which is going to be on the 14th of April. And that, unfortunately, is going to be me giving that talk, and that is going to be on alternative medicine, fact or BS. Okay, so it's partly tongue in cheek as well, but there is a very serious element to it also, particularly in terms of human health risk. Right, tonight's talk is being given by Professor Karl Ritz, Emeritus Professor in the University of Nottingham. And Karl is going to enlighten us about the remarkable world of fungi. Karl.
So I'm thinking I might do a cherry playground projection. So is that, can you all hear me fine? Or does anyone need the induction loop that this wretched cable requires?
Some over here can't.
You okay there?
No, I can't hear you very well.
How's that now? Is that better? Okay. How's that now? Okay, well, I think. Okay, so yeah, I'm Karl. I'm Emeritus Professor, which means that I'm now retired from the University of Nottingham, which means I'm a free agent, which is also why I'm smiling. No more students, no more bureaucracy and committees and all those sorts of things. I am a soil ecologist. By ecologist, which means that I'm interested in everything systems from molecules to whole landscapes and beyond. My background is I grew up on a farm in Oxfordshire. My dad was a farm worker, but I've always been a scientist. As long as I can remember, it's all I could ever be. And apparently, somebody asked me what I wanted to be when I grew up when I was 5, and the reply was, "I want to be a mushroom expert." So precocious little sod. But here I am, a mushroom expert, but much more besides. So tonight, I was offering talks to the Campus Scientific, and I gave a list of stuff that I could do. I was delighted that whoever it was chose fungi because they're kind of my favourite. So there's not much soil science tonight, but happy to talk about that at any other time, and lots of fungi. And really, I'm going to give you some background to this. So there'll be some natural history. So the first part talk will be natural history of fungi and so forth, but also as a research scientist and a cafe scientifique, it's a great chance to actually do a little bit more science talk than I normally do on outreach. So I'm going to take you through a few little experiments that I did with fungi, and you'll pick up the context there. So it'd be nice to do a little bit of some science on that as well. And so what I'm going to do is just set the timer because I do tend to go on a lot. I get quite enthusiastic, you might notice, and so it would be remiss of me not to start with something local and something that would be familiar. So I'm sure you're all familiar with mushrooms as an aspect of the fungi kingdom. And here's some nice examples of local fungal flora. All these mushrooms are within a 15-mile radius of Ulster that I've taken over the few years that have been down here since I retired. And yeah, already pretty remarkable, aren't they? This one here, of course, is the iconic mushroom. It's the fly agaric, and there could be a whole talk on that alone, actually. But I put that in because that's the one again everybody is kind of familiar with from childhood onwards, because this spotty mushroom appears in that early literature that many of us have been exposed to. So the key characteristics of fungi, in evolutionary terms, they emerged about 100 million years before present. So give or take, let's not be obsessed about that. So they've been around a long time, sure. But in classification terms, the fungi are a kingdom on their own. There are 6 kingdoms of life: plants, bacteria, archaea, fungi, plants, animals, and I've forgotten the other one. And very, very diverse. Estimates around about 75,000 named species, but there are estimates to be about 1 million species. So there's a lot that we don't know about. This is often the case especially with microbial species. Again, you'll also be familiar often with insects and things like that as well. The sort of scientific definition for fungi is from the Greek myces. So the discipline is called mycology, and fungal dudes are called mycologists. Fungi are very varied in terms of where they get their food sources from. We'll talk more about that in a minute. And their key structural polymer, what they're made of, is chitin. So, we're mainly the calciferous bones and things, and keratin, the skin. Plants, cellulose. Fungi, chitin. And what other organisms are chitin? Insects, yeah, arthropods. So actually, fungi are more closely related to animals, arthropods, than they are to plants. The Victorians have always lumped mycologists in with the botany department and this sort of thing. So actually a separate kingdom. But as always in evolution, these useful molecules are used in all sorts of different ways, and chitin is one that's common both fungi and the arthropods. And they are hugely significant, both ecologically, a little bit on that later, and also economically. And again, I'll touch on that in the talk shortly. Now, there are 2 basic morphologies of fungi, 2 body forms, if you like, for fungi. Fundamentally, we consider them to be microbes, but they're kind of weird, and I'll explain that in a moment. But this is sort of microbial end of fungi. Yeasts are a single cell form of fungus. They exist as these little round cells. They're about 3 or 4 micrometers in diameter. A micrometer is 1,000th of a millimeter, unimaginably small, even for microbiologists. It's crazy small stuff. And again, you'll be familiar with yeasts from bread making and brewing and everything else and so on and so forth. But basically, they are individual cells reproduced by just simply splitting in half. So one yeast cell splits into 2, 2 into 4, and on you go until they run out of space or food. So the yeast cells in these terms are the kind of the familiar ones, but also there are plenty out there in the wild. And here is a yeast again, just in the hedgerow near my village where I live, up near Bute. This is a sap yeast. So it's where the hedge has been cut by one of these horrible flail mowers. It's split the stem, so there's sugary solution comes out the broken stem there. And the yeasts, this yeast then is using all that sugar to grow this huge, slimy, rather spectacular mass. So keep your eyes peeled, and especially this time of year after hedge cutting, you may see some sap yeasts around there. And again, they are ubiquitous. So every leaf surface is covered in loads of yeast. And again, this is why when you're brewing, you don't need to add yeast. Natural yeast will take that up on that front as well. Now, the other form is of fungus we're going to spend a little more time on, but basically this other form starts as a spore. So we'll start very small and we'll go bigger, literally, as we'll see. So here's some nice range of examples of individual fungal spores. This is the basic reproductive unit of the fungus, and these are, again, tiny, a few micrometers. And again, they are ubiquitous, especially in the atmosphere. So they float around in the atmosphere and then get washed down with the rain and so forth. And each individual spore has the capacity to germinate and start growing a little thread that is called a hypha. So the spore puts out, I've got a laser pointer tonight, that's good. So the spore germinates, it puts out an individual hypha, which then branches progressively and extends by a process of apical extension, and then will branch and branch and branch, and on it goes. So if you follow that basic growth rule in so it's a spore, this is an example of an older fungus now, growing by these tips and branching. So what you end up with is a interconnected branching network, the technical term for which is mycelium. So mycelium is the basic other growth form of fungi, and it's much more predominant than yeast. There's much more diversity in mycelial fungi than yeasts. But the key features of this are that it is one interconnected network. All right, this is just a single organism, and this bit is connected to this bit, and the whole thing is all connected one to the other. And you'll see the consequence of that is that it can start to transcend scale. So this is a real little mycelium, one of my microscopic pictures, about 0.5 millimeter across here. But the basic growth rule is what we would call indeterminate. It can keep going, and indeed it does. So here's an example of, now with 5 millimeters, these are larger mycelia of 3 different species of fungi, and using a special technique I developed a long time ago. But again, you can already see that even though it's just a branching network, they have inherently different sort of morphological characteristics. So there's different sort of style of branching, and there's a different degree of curliness and these sorts of things. So already you can start to see that there's diversity in that basic growth form. So let's just have a quick look at the small scale. Here's a fungal tip. So this is the tip that grows by a process of apical extension. And nutrients are translocated down the tip and deposited here, and that's used to grow, and the cell wall then elongates. It's essentially also under hydrostatic pressure, okay? So it's always under pressure, which also then serves to drive the tip forwards. This is an electron micrograph picture. This is a fluorescence microscope picture. Here's a branch, and I've showed this because it also reveals that the fungus, the hypha is a tube, it's a hollow tube, full of cytoplasm, but also in some fungi there are little cross walls occur across these filaments. Some fungi have no cross walls, so that literally is one big bag, and some have cross walls, and some of the cross walls can be very complicated in terms of their structure. Again, we haven't got time tonight to go into all the nuances and consequences of that. But we can scale up. So here's an example. So I said there wouldn't be much soil tonight. It's a little bit of soil. Here's a fungal mycelium growing in soil. So it's a soil aggregate, soil pore, bit of food here, and this is the network. And you can start to see now how well adapted this growth form is for filling space. It's all about exploring space, because what the fungus is doing is growing through the soil, trying to find some food to grow on and grow more. So it's growing through the pore network. But already, again, you can scale extraordinarily large and strikingly complicated. But it gets more interesting than that, because some species of fungi, the individual hyphae will aggregate together to form what we call cords. So if you get a bunch of hyphae that come together and form a cord, this is a cross-section of a cord, lots of individual hyphae, they can fuse and hollow out. And basically what they're doing is forming a cable. And of course the cable is 1 or 2 orders of magnitude larger than the individual hyphae, so it could start to transcend scale much more. Now we're on 5 millimeters, and here's a cord-forming fungus growing across the soil surface, looking for food, and it's found it because this is a piece of straw. So this fungus is foraging, it's exploring space to find food, it finds that space, and it proliferates on the straw and decomposes it and eats it, and then can carry on growing off into distance. And so, again, this is the way that the microbe can transcend scale, by aggregating together. And it gets crazier than that, because there's my hat, which is under the table here. And this is a fungal mycelium growing in a rather uniform and rather boring sports field. But it's just like the little collies I showed you to start with. It's started in the center here, it's growing out. So this is the growing front of the fungus. Fungi, they get their nutrients by pumping enzymes out that digest material outside the fungus, and they absorb it. And of course, that's a very messy way of eating. So the nutrients that are released, the fungus does not take up, are then available for other organisms, and in this case, the grass, which is why you get the green ring. The fungus is not growing in the middle here because it's been through there and eaten all the stuff it wants. So it carries on growing and can get larger and larger and larger. And prosaic old mycologists and professors of mycology still refer to these as fairy rings, which is rather lovely. So you get erudite scientific papers talking about fairy rings. And again, depending on the season, etc., then you could actually get the mushrooms. Mushrooms can appear, so you get a champignon, fairy champignon ring like that. And again, all sorts of other things going on. underneath there. So we're now into what, many centimeters, but it gets even crazier because basically the fungus is it can keep on going. So there are many documented examples now of very large fungal colonies growing, especially in ancient forests. And here's an example of a mushroom fungus called Armillaria bulbosa. And this is essentially a map of the extent of mycelia in this forest. A clone is a genetically identical individual. So the ecologists here sampled the soils and detect the presence of this Armillaria, this clone and this clone, and they mapped them out. And something suspicious is going on here because I put a compass on there. And astonishing, the ruler is 100 meters. So these organisms are individuals and they are hectares in extent. So the largest organisms on the planet is not the whale shark, it's fungal mycelia. And again, the view is that these are very, very ancient organisms. The other starting thing, of course, is that they are connected. This bit of the mycelium is connected to this bit of the mycelium, and they kind of know what's going on within that individual organism. Again, a profound sub-story that we haven't got time for tonight. So, fungi are considered to be microbes, but they're also macrobes. In fact, they're the biggest macro that you can get, and everything in between. That's remarkable. Remarkable thing, number one for this evening. So, what can you do with a hypha? I'm going to show you some remarkable things that you can do with a hypha, including making mushrooms. So mushrooms are the reproductive structure of a particular form of fungus. In taxonomic terms, they're called Basidiomycetes. And the mushroom is actually just mycelium. It's just hyphae that are knitted together. This is an example of a shaggy ink cap. And here's the cap. If you zoom in on that point, which you can do, if you find one of these, you can see. Look, you can see the individual threads. So this is just a bunch of mycelia. They're aggregating in a particular way to form this cap, and you can see all the strands and threads there. So this basically, it's fungal knitting that creates the mushroom. The mushroom is a device that produces spores, and I'm going to show you a few more sort of more outlandish fungi again, just to begin. Remarkable what you could do with a hyphae. There's a whole range of fungi called stinkworms. These are phallus species. Their spores are produced as a sticky mass on the tip here of the structure, and there smells of rotting flesh, which attracts flies. So the flies come and eat this, and then they carry the spores away. This is a British stinkhorn. This is a tropical one. And I put this up again because I think it's some evidence that evolution has a sense of humour. And also, how outrageous can you get in sort of form from mycelia? So again, these are tropical fungi. We don't have them in the UK, but they're really quite stunning. So the species, they're allied actually to stink. They rely also on insect dispersal from smelly spore clusters. But you can imagine, devil's fingers and the cage fungus, really just quite extraordinary structures. And again, get deeply embedded into folklore around. Yeah, pretty amazing. So let's move on to reproduction a little bit. Fungi reproduce by spores. Again, that's we've introduced that so far, so familiar. But they will also reproduce by fragments. So if a fungal mycelium gets smashed up and falls into lots of pieces, little fragments, each of those fragments can regenerate and grow a new mycelium from that. And that's vegetative reproduction. The fungi that can't do that are the ones that haven't got any of those crosswalls in. So there's an example of why the crosswall can be important. So those fungi that have crosswalls cannot reproduce by fragmentation. They're very prone to being damaged in those terms. And again, I'm sure you're all familiar with a mouldy piece of fruit. Often penicillin is the archetypal one here. This is this green fungus, and it's growing really prolific. There's so much sugar in there, it can produce loads and loads of mycelium inside the fruit, and there's millions and trillions of tiny spores on the surface here. Down the microscope, they look like this. So the penicillin has a little stalk, and then a bunch of subhyphae, and the spores come off here one after the other, and they get carried off into the air currents and carried away. And again, there's a huge diversity of the morphology of fungal spore structures. This is the classic penicillium one, a bit like a hand. And here's another rather lovely one called Aspergillus nidulans, which is a really prolific. You think of the number of spores that are there. This is one, 10 micrometers. So there's thousands and thousands of these structures piddling out all these spores. So hyper prolific. And also, this is why the atmosphere is so full of them. We are breathing them in all the time as well. But because we've done that since the dawn of time, our immune systems are able to cope with by and large, with moderate loads of fungi going in there, unless, of course, you're immunocompromised, and then it gets much more serious around that. And so the mushroom, as I said, is a spore dispersing structure, and the way that works is that here's an example of a wax cup, and I chose this because it has nice big gills, so you can see the gills underneath it. These are like little plates that hang down within the umbrella shape of the mushroom. And again, next time you're cooking with mushrooms, but you've got open cap mushrooms, have a close look and look at the gills, because the surface of the gill is covered with these tiny, tiny structures which are producing the spores. There's little sort of base structure, and then in these sorts of fungi, there's always 4 spores on the end of each of these bases, and these spores fall off. They get spat off the end of this structure. So perpendicular, they fall down. So you have the umbrella of the fungus there. The spores are falling down from the gills, and then they get carried away by air currents that are running underneath the fungus. And again, some biophysicists have looked at the shape of different mushroom caps, and you can model airflow. Some of them are shaped to optimize making an uplift current as the airflow goes across the cap as well, so that spores are going to get dispersed more effectively. Other tricks. This is not a basidiomycete. This is a different sort of fungus, but rather than having gills and caps, it's a puffball. So these are spherical fungi, but the spores are produced inside here. And again, when you're in the woods and you see the puffballs, notice that they're often occurring around the edge of trees or on the edge of the woods, because that's where the raindrops are falling. And it's the raindrops that come down and hit the puffball and puff out the spores. So it knows where the raindrops are, and it knows then where to produce the puffing structure around that. And there are, again, so many other different dispersal mechanisms we haven't got time for. But so what do they do? This is sort of the basic biology of the fungus. But what do fungi get up to? Well, in ecological terms, their principal role is that of nutrient cycling and decomposition. If we didn't have fungi decomposing stuff, especially organic stuff, we'd all be up to our, beyond our heads in rats. And of course, that decomposition also just drives the nutrient cycles that make the systems work. And so, they are highly adapted to decomposing organic material. So here's one growing on a cow pat, and then here's the archetypes of wood decomposing fungus. So it's the fairy bonnets decomposing this wood. And what they're doing is the mycelium is inside here, degrading the carbonaceous substrates in there and using it to grow and releasing stuff, and on they go. And again, kind of whatever it is that's organic, there's a fungus that's going to decompose it. But they're also involved in decomposition of inorganic materials. And we'll meet another one in a minute, but I want to start with this one because this is a remarkable one in the sense that these are the fungal hyphae growing in and decomposing this material. You can see they're drilling their way in through here. They've been staying here so that we can see that they're working their way through. And this matrix, astonishingly, is a tooth from a medieval graveyard. So, and what the fungus is doing is drilling in here to get the phosphorus, because there's lots of nice phosphorus inside of it, and so it's able to get in by it exudes acids that decompose, degrade, and it can drill its way into this tooth. And So, fungi are busy there decomposing archaeology, and many archaeologists do not appreciate that. A lot of arguments with archaeologists but the other inorganic story that's important here is we can start to also just pick up the narrative in terms of how fungi interact with other organisms. So fungi in evolutionary terms have combined with other organisms to create some rather special structures. And the principal among those in evolutionary terms to start with were fungi connected with algae, which are individual, they're microbial photoautotrophs. They're green, little green cells that can fix carbon. So they hooked up with fungus and produced lichens. So lichen is actually a fungus and an alga living in symbiosis. And what happens is that the green plant fixes the carbon, energy from the sunlight, makes sugars, it passes on to the fungus grows, lots of hyphae, and provides a matrix, the green stuff, to hang around it. So it's like a proto-plant in that sort of sense. And lichens are really, really important in terrestrial systems because, again, they are common on rocks and so forth. And what they're doing is they're actually degrading the rock material as well. So the fungus is exuding acids to get the phosphorus out of the rock, and that's eroding the rock and it falls off, and that's all part of the very early stages of soil formation. So I haven't got time for the rest of the soil story, but here it is at the very beginning. And of course, lichens have also adapted as all life to other habitats. So again, you'll be familiar with them, especially around here because there's nice clean air of lichens on tree surfaces and so on and so forth. The other place you'll find fungi ubiquitously is on the outsides of buildings. So this hotel, all the stonework throughout walls, whatever else, those surfaces are certainly supporting a wide range of fungi. They're hanging on in there, doing a little bit of degradation, and picking up nutrients, in lots of nitrogen from the cars going by nowadays, etc., etc. So truly ubiquitous, not just decomposing organic material, but making a living in inorganic circumstances as well. And the other symbiosis that we need to talk about is where fungus connects to plants. So not so much micro plants of algae, but actually higher plants, multicellular plants. And again, this occurred very early in evolutionary terms. And again, nowadays, I think many people have actually heard about mycorrhizae. 20 years ago, this was revelation to people. So I'm not going to spend too long on it. But basically, the way the story goes is that there are a whole range of different sorts of mycorrhizae, 5 or 6 sort of primary sorts. And basically, the fungus will connect to the plant root. And here's an example of a pine tree with this fungus, Suillus bovinus, connecting to the roots and growing off into the matrix. This is an experimental system set up by a mycologist to demonstrate. But all this branching stuff around here is not plant root, it's fungal hyphae. And in fact, these are cords. They're so large you can see them. These are cord-forming fungi as well. So what it's doing is the plant is giving carbon to the fungus, and the fungus is growing through the soil. But because the fungus is essentially microbial, for a given amount of carbon, it's able to grow through a much larger volume of soil than the plant root will be able to do. Okay, so what the plant is doing is it's investing carbon into a microbe, and the microbe can get through a lot of a larger volume of soil than it would be able to do otherwise. So the fungus grows up into the soil and picks up nutrients and water and pumps it back to the plant. So the fungi are essentially an extension of the root system. And the vast majority of plants support mycorrhiza. Only the cabbage family does not have mycorrhizas. All other plant species have mycorrhiza connected of different sorts. These are called, the ones that connect to woody plants like trees are predominantly called ectomycorrhiza because the fungus, when it connects to the root, is predominantly on the outside, forms this huge proliferation on the outside of the root, like a mantle, it completely covers. So this is a fungal mycelium, and the plant root is inside there. And then there's also those cords. This is just what we call the mycorrhizal tip. There are also cords going off into the soil, doing all this transport stuff. These are the ones that are common on woody species, and they're on the outside. All the herbs and the other plants have a different sort of mycorrhiza called endomycorrhiza, and endo is because the majority of the fungal mycelium is not on the outside, but the inside. So this is an example of, now again, in real soil, this is one of my pictures of plant root coming down. These are root hairs, these individual ones, but they're branching much longer structures are the mycorrhizal fungi growing off into the soil. A root hair will typically be no more than 3 millimeters, and already you can see this fungus is growing off into the distance there. So orders of magnitude exploration of that soil volume. And the endomycorrhiza then, it works like this. The subterm is arbuscular mycorrhiza, and we'll come to why that is in case in a minute. But here's the fungal hypha coming down, and it's forming a little infection point. This is the root. This is the surface of the root. It's formed a little infection point, and it's grown inside the root. It's a zooming away from this. So here's the hypha coming. This was out in the soil originally before I washed it out. There's the mycelium. There's the hypha coming in. There's the infection point, and it's the very, very early stages because it's just infecting here. This is the cortex of the root, and there's the waterworks down the middle. And what happens is the fungus grows through this cortex and proliferates. And again, in some species, to a really considerable extent. So here's the fungal mycelium inside the plant root. And in fact, you can see that there's not a lot of root left there now. It's merely all fungus. Yeah. And then eventually, some of the fungi, some of these endomycorrhizal fungi form what we call vesicles, which are little balloons full of fat that the fungus uses to survive should the root die. So it allows the fungus to carry on proliferating for a while. But in the vast majority of cases, roots out in the real world are not roots, they're mycorrhizae. Okay, this is the normal circumstance. And what it does is it gives the plants a foothold into that source in terms of nutrients. Now, the transmission of the carbon to the fungus and, say, the phosphorus that the fungus has picked up to the plant occurs in a structure that is inside the plant cell, and it's called the arbuscule, which is why they're called arbuscular mycorrhizae. So this is an example of an arbuscule growing in the root. Here's the hypha coming down. This is a single root cell, and the fungus is branching and branching and branching. And look how fine those branches are. Remember my subtitle was branches everywhere. Well, we've had hectares of branching down to 10 but less than a micrometer a branch going on. And it's this shape because it's filled the entire cell. And arbuscule is Greek for little tree. So indeed, when I was out on this weekend, I thought, I need to do this. Look how similar. Look how nature solves the problem of filling space. Okay, so very, very similar. Now 1 meter rather than 10 micrometers. Okay, so you can have fun when you look at the hedges in Devon Cornwall, arbuscular type trees. And then the couple of quickies on the effect of mycorrhiza on the plant growth is that principally it's about nutrition of the plant. So as I said, it extends the root system so that plants are able to get water and especially phosphorus from the system. Phosphorus is an important part of the story because it moves in soil so slowly. So that if the plant is relying on diffusion to get phosphorus to it, it very soon will absorb the phosphorus from the root surface and then it will run out because the concentration gradient will decrease. The phosphorus can't diffuse fast enough to the plant, so the plant gets hungry. But the mycorrhiza can go out and pick up phosphorus from a long way away and pump it back into the plant. And it's one of the earliest deeply scientific demonstrations of the impact of mycorrhiza on plant growth by pioneering scientist called Barbara Mosse. She was a character, I tell you, back in 1969, where she was growing onion plants that she inoculated without mycorrhiza or added mycorrhiza, and you get straight away you can see the difference in the size of the plant, and it looks like an onion, it doesn't look like an onion. And the reason that onions was the starting point to explore this is because, again, perversely, Alliaceae, onions, leeks, yada yada yada, do not have root hairs. So they rely enormously on mycorrhiza to provide the substitute for a root hair around this. And that's always in biology, the complete variety of every circumstance you can kind of think of. But yeah, so onion roots are bored, and they have to connect with the fungus to get going. But the other thing is, so this is the impact on an individual plant, but they also have a profound role to play in terms of interactions between plants. And again, and forgive the indulgence, but I wanted to show you something from my PhD 46 years ago, where I was looking at the role of mycorrhiza in plant-plant interactions in grasslands, especially in terms of nutrient transfer between plants. But basically, what I did here was I'm growing some ryegrass plants with some plantains, ribwort plantain, something like that. in pots with a screen to separate the shoots, so there's no competition between the shoots. And here's a ryegrass plant and a plantain plant in soil without any mycorrhiza. And then I put mycorrhiza in this treatment, okay? And what you've got is now you notice big grass plant, small plantain, big plantain, same size grass. So what it's allowing is that the plantain can compete for nutrients and phosphorus with the grass when mycorrhiza are present, because the fungi give it the ability to get at the phosphorus that it can't otherwise do, because the grass roots are much hairier than plantain roots. So you put a fungal hairy system in, and then they can coexist much more equitably around that. So fungi affect the interactions between plants, and there's all sorts of community dynamics consequences arising from that. But what it can also mean is that because some of these fungi are quite gregarious, they will infect a number of different species. So as well as infecting, plantain to plantain, it can affect plantain to ryegrass. And these form structures then, there's a possibility, what we call common mycorrhizal networks, where a mycorrhizal network can come and join plants together by this common mycelium. And this has got really, really trendy again over the last few years. It's become much more so. You've probably heard of the wood wide web and this great harmonious interconnection of all plants and each plants, and they talk to each other and all this sort of stuff and yada yada. And that's really got rather hijacked because push comes to shove. And again, I was researching this 46 years ago, and I could find no evidence actually for enhanced nutrient transfer between plants in presence of the mycorrhiza. And if you go, this is the Café Scientifique. If you go hardcore and look into, this is a couple of recent papers looking and reviewing about this, and you'll see from the title, positive citation bias and over-interpreted results lead to misinformation on common mycorrhizal networks. Systematic mapping of experiment approaches to study. The conclusions are the claim that mature trees preferentially send resources and defense signals through common networks at the present has no peer-reviewed published evidence. We feel that not all studies tested networks formed exclusively of mycorrhiza were present that meet the stringent definition to sort all this out. Okay, so caution on this rather attractive concept. Again, happy to pick that up for a further conversation if you like. So there's some symbiotic interactions between fungi and other organisms. There's also a bit of antagonism goes on. Again, it's not all harmony out there. It's a sort of trend in ecology at the moment to make it all lovely, but no, there's plenty of pathology going on as well, and there are many fungal pathogens that attack all sorts of organisms, plants most especially. So here's some real serious suspects. Yellow rust on cereals. The Romans had a lot of trouble with this. In fact, they used to sacrifice red dogs to try and control yellow rust. Rice blast, a lot of starving people as a result of rice blast in that grass. Maize smut, I put in because it's spectacular, where the seeds of the maize become substituted by this smut fungus that expand. This is now a black mass of fungal spores, using all that lovely starch from the sweet corn grains. Apparently, in the Americas, maize smut is a culinary delicacy. I think it's called beet nakosh. Anyone eat beet nakosh? Is it good?
Yes.
Right. It wasn't on the menu tonight. I looked. And then potato, right to Solani, black scurf and stem canker again, quite a tricky disease. And I put that in because I'm going to show you some experiments involving that in time here. Okay, good. So as well as plants, fungi affect animals, and again some pretty spectacular examples here with some invertebrates. Chitin meets chitin, yeah. On insects, so here's a fungus erupting from this fly. Here's a cricket, and then arachnids. There are a huge range of fungi highly adapted to killing and eating arachnids. So here's mycelium spores coming out of the spider. This one is this is a tropical one, and again, a wide range of these. These are called zombie fungi. You can Google that if you want to sleepless night. But here's the spore structures coming out, and very high up to get to the air currents. Not much air currents in rainforests, so these always have spectacularly high spore energy around that. And then nematodes, again, little soil organisms. These are the most ubiquitous animal on the planet. That's again, that's the soil ecology story for another day. But nematodes are tiny wee worms that are ubiquitous, but lots of them in soil. And fungi, there's quite a few fungi that are adapted to capturing and killing and eating nematodes. And you might wonder, well, how does the mycelium, how does the filament, how does the hypha deal with this wriggly worm? And the answer is by a wide variety of ways and astonishingly effective. Amongst them are nematode superglue. So this yellow fungus, here's the hypha, here's the stalk. This is not a spore. This is a blob of nematode superglue. So this is in the soil pore. The nematode goes wriggling through the pore, hits this and gets glued on. So here it is, stuck on here, and it can't wriggle away. It's actually glued on. And then the fungus will penetrate the nematode and grow through and gobble it up. The second strategy that they can use is they make lassos, snares, essentially. A snare is a very effective physical device for catching stuff. As you know, if you trip over the hose in the garden. And here's the individual hyphae, and they form loops with 3 compartments. And these compartments contain membranes, and as discussed earlier, they're under a hydrostatic pressure. So that what happens is that if something goes through the loop, it's detected and triggers and causes a rapid expansion of these 3 little bags of water, and they pinch together. And what happens is the nematode comes crawling through the soil pore, and being a noose, it puts its head through, and the bags expand, and it's been trapped. And you notice that there's 3, which of course is the ideal number for a guaranteed uniform pressure and optimum capture mechanism. You might go, yeah, come on, how likely is that? But if you think about it, this is in a passageway in the pore. It's going to happen. And here's an example of it happening twice, not in a passageway, but just in a culture system. So both head and tail has been caught by this nematode. So one bit must have got caught and wriggled, and then it went into the other loop, and there it is, captured. So remarkable, eh? Right. Just going to move a little bit onto some experimental things. Again, so again, a little bit of natural history. I just want to do some experimental stuff with you because, as I say, as an experimental scientist, and it's a scientific meeting, so a chance to show you some stuff. And as you know, I'm a soil ecologist, so one of the issues facing microbiologists is that what the thing that I call cost legacy, which is where microbiologists tend to look at individual organisms growing in boring old culture media. It's an agar plate with fungus growing on it. And this is just a sensory deprivation chamber for the fungus. Because in the real world, it's not like the substrates and the environments that fungi grow in. They're much, much more complicated. Here's soil with some roots and some fungi connecting, so on and so forth. So what I wanted to do was start to understand how fungi respond to this sort of environmental heterogeneity. Because again, most mycologists have just not thought about these things. They're just working with stuff in culture media and so on and so forth. So I started to think about this and wanted to start to research. Basically, the question I was asking was, how do fungal mycelia respond to heterogeneous distribution of nutrient resources? Okay. So what I did was I took the concept of the agar plate, which is uniform, and chopped it up into little cubes. So I could have little cubes of agar jelly that were uniform in terms of their nutrients, or arrange them in patterns of high and low nutrients. So this is the equivalent of a standard agar plate, all uniform. And then you can play around and patch them up like this. So you now have a controlled heterogeneity. You can put fungal spore in the middle and see what happens when the mycelium grows off across. So they can bridge air gaps. You've seen that in the other pictures. How do the fungi respond to these different circumstances? So there's an example of the fungus growing from a high nutrient zone to a low nutrient zone. You can see all the hyphae going across. Look at that for adaptation to filling space here. And you can see straight away that it's prolific in the high nutrients and it's sparse in the low nutrients, this particular species. You have all sorts of fun mapping and playing with these patterns and looking at the responses. So this is the pattern of nutrients. This is the extent of the fungus. This is how far it grew. And this is the density of the fungus. What you can see is that for the low nutrient system, the fungus colonized the entire area very, very quickly. But in the high nutrient system, it colonized these adjacent tiles and didn't bother with the peripheral tiles. But the density was reflected like that. And then the patterns are reflected in all sorts of different ways around us. So what I started to do was to understand that actually the mycelium is spatially adapted to the nutrient distributions there. But also there are differences between different species. So again, there's a wide variety, diversity in the way that different fungi respond. So here's that potato package from Rhizoctonia solani growing in these systems, and these are now photographs. This is the nutrient pattern. Here's the fungi growing on these gels. What you can see, you can't see the hyphae here because of the scale, but the dark stuff here is reproductive structures that this particular fungus produces. What you notice is that it always deposits its reproductive structures in the low nutrient zones, not the high ones. This is high, this is low. So it's depositing its reproductive structures in the zone where there isn't any food. What that suggests is that it's because it's run out of food, it goes into reproductive mode at that point. But the other thing is it's asymmetric. It's wandering all over the place, and there's no reproductive structures formed in these low nutrients, only this one. So some fungi are very symmetrical inherently, and some are very asymmetrical inherently. Again, all sorts of ecological consequences arising upon that. And again, you can extend that concept and look at so cutting those little squares took my PhD student ages. So we devised the faster system where they were she just put out drops onto the surface. And this time, what I wanted to look at was a functional response rather than just the growth response. So what you have is a bunch of little agar spots as before. You inoculate the middle, and you can fiddle around with what's in these gels. And this was looking at the solubilization of phosphorus here. So some of these dots contain phosphorus, contain phosphate, and some contain sugar, contain glucose, and some contain both. So you can play around with all these different patterns. I'm not going to go through all the nuances here, but we'll stick with some simple examples of just some examples of no glucose. So this is just a super hungry fungus, okay, and you can barely see the hyphae one day or even 7 days later. We'll have later. So this is like the control, and then if you have it all full of phosphorus, you can see the grains of phosphorus. It's an insoluble form of phosphorus. Again, not much is happening. Okay, so those are like the two control treatments. But if you start mixing and putting glucose and phosphorus together, so the fungus has got both, then after 24 hours, not a lot's happening. But after 144 hours, the fungus has grown profusely. You can see all the mycelia here have grown out and around this. But the other thing is that the colour of the dots has changed. And that's because we put a pH indicator in here, so that when the phosphorus has been solubilized by the production of acids from the fungus, the colour will change from orange to yellow. So what's happened is the fungus has acidified these zones, turned the indicator yellow, and solubilized the phosphorus so the gel has gone clear. Remember the lichens in the tooth? So this is in miniature going down. It's uniform, okay? Uniform on board. What happens if you go heterogeneous? So now you put the glucose in the middle ring and the phosphorus in the outer ring. And after 24 hours, the fungus has started, you can see some little hyphae here, just started to grow out and find out, but since rather later, okay, colonized the whole area, and the phosphorus has been solubilized in the outer ring by the production of acids from the fungus. So the fungus, there's no carbon in this outer ring, okay? So what the fungus has done is it's taken carbon from this inner ring, taken the outer ring, and used it to solubilize the phosphorus. So it's spatially organized and sorting itself out in terms of moving stuff around where it needs it. If you reverse that, so you have phosphorus on the inside and glucose on the outside, then the fungus trundles across, grows into the outer ring, gets the glucose and grows furiously and starts growing out there. But what's really curious is that it's also solubilized the phosphorus on the inner ring. So whilst the fungus is actually growing in both these directions, it's translocating the material backwards as well. Okay, so again, it's back to this business of a coherent, integrated network that can spatially organize around according to the circumstances it finds itself in. This was pretty pioneering stuff back in the day. So the next quick experiment was, again, just a little bit of tiny bit of soil science here. And I just want to put this one in to give you sort of one step forward in one of my other research interests. So what I wanted to do was sort out how the biology in the soil affected the ability of fungi to grow through the soil. Okay, tricky question. Experimentally, how do you deal with that? And I had a Japanese postdoc came and spent a year with me. We did some fabulous work here. We take little soil experimental system. We take little soil aggregates and stick them in a petri dish. This is a cross-section petri dish. So 3 aggregates in a row on little rubber pads. So they're isolated, okay, spatially isolated. And then you could treat these aggregates in different ways according to the questions you want to ask. And basically, I'll come to that in a second, but you put a fungus on the middle one and look at the ability of the fungus to go across the middle one and colonize these adjacent aggregates. So here's an example of where I put a load of sugar in the middle, the fungus is proliferating on there, grows across and grows into this aggregate. Okay. And what we did was we used this to study a number of questions about the role of other soil organisms in affecting the ability of the fungus to grow through the soil. So we treated the outer aggregates in different ways to affect the biodiversity in a general sense of those natural aggregates. So the question here is, what's the impact of biodiversity of the soil on the ability of the fungus to colonize that soil? So experimentally, we manipulate it in different ways. Don't worry about all the nuances there. But the point is that you move from a sterile aggregate, okay, no life in there at all, to a non-sterile one from the field, which is full of life. And by fiddling with different systems, we can make a diversity gradient in these. And our hypothesis was that the more diversity, the less able the fungus would be able to colonize the adjacent aggregates. Very important to set the hypothesis up before you do the experiment. So we ranked these treatments. So this is the gradient that we thought would happen. And what we see is that there is indeed a gradient of ability of the fungus to colonize contingent on the diversity. So the sterile system, you get 5 units of fungal colonization. The non-sterile system, 3 units. These are log units. 2 orders of magnitude less ability to colonize, sterile versus non-sterile. And then different diversity levels, there's a gradient. Okay. So the more diverse the system, the more difficult it is for fungus to grow in there and it's an invader. If that's a pathogen, that's important, yeah? And the other way was to start looking at more again, this is the joy of experimentation. You can manipulate things. We sterilized all the adjacent aggregates and inoculated them with different bacteria, individual strains of bacteria. So a little bit of Koch's legacy stuff here, but each bar is a different bacterium. Here's the sterile, of about 5 units. Here's the non-sterile, okay, same response, 2 orders of magnitude less. Different bacterial species attenuate the ability of the fungus to colonize to a different degree, and one of them was as effective as the non-sterile system. And this particular bacterium was chosen because we knew it was a strong antibiotic producer. So bacteria can produce antibiotics that will inhibit fungi, as well as fungi producing antibiotics that can inhibit bacteria. As always in nature, things work both ways. And the same thing with fungi. Essentially, pre-colonize the adjacent aggregates with fungi, different species of fungi, and say sterile, loads of colonization, non-sterile, very little colonization, a whole raft of different abilities. So it kind of depends who's in there, whether this particular fungus can get in. And putting this into an ecological context, the hypothesis was that this was about niche occupation. So that if there's high diversity, then all the niches are occupied in that soil, so that the organisms can't get in there. And this allows us to test this hypothesis because, lo and behold, these 3 species are other Fusarium fungi. Okay, so our test organism was a thing called Fusarium oxysporum, I said at the beginning. So the Fusarium has difficulty colonizing adjacent aggregates if other Fusaria, different species, are in there first, which suggests that there's a Fusarium-shaped niche in the soil. If it's occupied by another Fusarium, then the other ones can't get in. And again, that has all sorts of biocontrol implications. So some 3 very quick snapshots of some little simple, straightforward, dead cheap experiments that can tell you all sorts of interesting things. Now, there's probably some of you thinking, oh, well, that's all real. You're an ecologist. It's all a bit woke, all a bit easy speedy. It's all very, very qualitative, arm wavy stuff. So just to point out that mycologists, we also do heavy duty science as well. And I've worked a lot with mathematical biologists modeling fungal mycelia and fungal systems in all sorts of ways. So we can do some heavy duty quantitative stuff as well. And picking up things like this. And also, if you're into stochastic modeling versus differential equations, all this sort of stuff, then I've also used it to look at spatial community dynamics. The big issue with fungal mycelia is that they interact with each other when they meet. That's a complicated thing to study. So we've used some stochastic approaches and made analogous with my agar tile system using cellular automata systems, for those of you who know about these sorts of things. We've been using automata to model community dynamics, which again gets a bit heavy and not really suitable for evening after dinner. So finally, I promised you in the abstract a little bit of human fungal interactions. We haven't got time to go into that in a lot of detail, so it really is just a teaser. But we've talked about fungal plant interactions, fungal animal interactions, fungal human interactions. There are human fungal pathogens, ringworm, it's not a worm, it's a fungus. Thrush, sorry ladies, that's a fungus. And the virulent form of thrush is when it transforms from a yeast form into a mycelium form. So when it's a mycelium, you've got your trouble. So again, so medical approaches here is that you need to stop, you have to understand how yeasts transform into mycelia, and then you can start to control thrush. And again, that's what mycologists have done, understanding the molecular basis of those transformations. So again, I don't go into details, but basically, human and fungi in terms of agriculture for plant productivity, food, yeah, for sure, medicines, antibiotics, vitamins, biotech, all sorts of very interesting things going on in biotechnology terms now, with fungi in terms of producing biomaterials, enzymes, biofuels. And in research terms, again, phenomenally powerful systems from genetics perspective, but also in terms of networks, very interesting. You can use fungal mycelia as a model for understanding the origins and consequences of network phenomena in systems. But it also connects much more deeply. Humans and fungi go back a very long time. So true ethnomycology, here's some Saharan rock paintings from 8,000 years before present, and there are distinct mushrooms. There's some covered in mushrooms, and here are some mushroom-like figurines. So again, mycologists and ethnomycologists think there's all sorts of important stuff going on back there. Of course, that translates even into Victorian times with the Alice in Wonderland, folklore, gnomes and fairies, and all that sort of stuff. And then, of course, the really nice end is where you move from fungi's food to fungi's Epicurean systems. So truffles, and again, the interaction of mammals with fungi. Mammals, dogs, mammals, humans. This is a beautiful Lagotto hound that they use for detecting truffles in Italy. I had a lovely time lately. I'm a very doggy person as well, so that resonates enormously. Okay, so the other one is just remember the how many $1 trillion of plant pathology cost. Just to put into context. Really nice report from Kew, State of the World's Fungi 2018. Well worth looking at in PDF download, but they pick up, then they were estimating the global market for edible mushrooms alone is about $42 billion a year. And in terms of a food source for humans, fungi have so much more potential than is currently being realised. And again, that's a bit of a hobby horse on my side. So to finish with, well, the fungal kingdom, pretty remarkable what you can do. The simplest thing of an extending thread that branches. You can produce these mycelia that do all sorts of incredible things in experimental systems, but also out in the wild. Mm-hmm. This is my current favourite fungus picture of a shaggy ink cap dropping off a gooey mass of black spores that comes. It's very popular right now because the rain is washing the spores down into the grassland, but I think that's really rather lovely. Thanks for your attention.
Thank you so much for that, Karl. That was really fascinating. When I was at university over 60 years ago, doing biology, we were taught mycology as part of plant science. And just shows you how much it has changed. But I often wonder what the socioeconomic implications might be for those vegetarians and vegans who enjoy their fungal protein meals without realizing that fungi are much more closely related to animals than so that might be something to watch for the future.
Also means that eating arthropods is fine.
Yeah,
It's just fun. It's just a leggy bun.
questions for Karl, please.
Okay, Carl, do you use mushrooms in any form additionally for yourself?
Personally, I use them for culinary reasons alone, because as you can see, I'm so high on life anyway. I don't need things to make me hallucinate. But from the medical perspective, so again, from the hardcore pharmaceutical side, so prescription stuff, so I personally do not partake of other forms of mycopharmacology. I do know people who do, and the apparent efficacy is, the evidence for efficacy, etc., is increasingly strong. What we need is a considerably more liberal system in terms of research and application, and I would, fully endorse that. Pharmaceuticals, pharmacology really needs to get their head around this a lot more seriously.
It's all about money with that, isn't it?
Yeah, I mean, it's a whole different conversation. This whole business of the industrialization of society and the consequences of that. It could actually be a whole kind of scientific session. Yeah,
I would say that I've been using mushrooms medicinally for myself, which is in my food for 12 years, and it changed my life.
Yeah,
and personally, yeah.
So, but of course, all of these things always come with a caveat. Carving knives are dangerous. Use carefully. Mushrooms can be dangerous. Use carefully. It's all risk assessment and common sense. But yeah, we'll for it.
I'm absolutely intrigued that cabbages don't have this involvement.
Yeah.
Kind of thinking, why cabbages? Presume the whole brassica.
Crucifery are non-mycorrhizal. Why? Well, so nature is a strange construct. So they can cope without mycorrhiza for a number of reasons. One of which is they're extremely hairy, and they have amongst the longest root hairs. They're also very good at producing organic acids, which can mineralize phosphorus. They are also, generally in ecological terms, pioneer species that do not have to deal with all these other plants competing with them. So they tend not, correct me if I'm wrong, I can't think of any crucifers that you find in dense natural grassland, for example, where it's so much competition going on. So the ecological answer essentially is that they are occupying a particular niche that they are adapted to and can fit into. There's a whole bunch of stuff that crucifers can't do, which is their trade-off for occupying that niche. Yeah, it's like it's this big natural jigsaw.
Yeah. The cabbage families produce a lot of brominated organic compounds as well.
So that would be competition.
And they would be toxic fungi.
Well, they'll be toxic to some fungi, but not others. So again, as always, there'll be fungi that are adapted and they deal with. So there are a range of fungi that can degrade glyphosate, no problem. There are a range of fungi that are extremely susceptible to glyphosate.
And sulfur compounds.
Sulfur compounds, yeah. So sulfur is the classic. So sulfur is a classic fungicide. It's used. You can use sulfur, but there are some fungi that don't mind a bit of hydrogen sulfur. It's all biochemistry thing. And again, biochemically, fungi are hyper-diverse. Okay, they just knock all other organisms to a cocked in terms of biochemical diversity. Again, fungal biochemistry is just the number of secondary compounds they produce is kind of beyond imagination, which is also why they have such an application in biotech terms.
Yep.
I've come across some interesting cases where the fungi are actually growing in materials which will kill them by growing by growing in the solvent and absorbing water. Yet if you actually put them in water and the chemical, it kills them. And yet they're actually growing in drums of these chemicals, which is quite spectacular.
It's their crazy membranes to deal with this.
And it wasn't just one chemical; it was more than one. The other interesting thing was you actually raised it with thrush, is that many yeast I come across actually grow in fermentous forms, particularly in presence of bacteria under stress conditions. So I frequently find yeast growing in not almost mycelial form, but they're definitely yeast.
So again, very interesting observation is that you're noticing that when bacteria are present, so it's back to this business of growing microbes on their own, you don't know much about them when you put them in the context of other organisms.
Yeah.
Other things happen. And these yeast, we call this dimorphic form. So it can be a yeast-like or a filamentous-like, and you can switch between. Dutch elm disease is another example. So Dutch elm disease in the as an exploring phase of the mycelium, and when it gets into the plant, gets into the tree, it switches to yeast, which then grows up through the waterworks in the tree and clogs it up and basically chokes the tree out. So it's the reverse to thrush.
So the other issue with the yeast was them forming spores, and again under stress conditions when there was very little nutrient. We're trying to identify why it happened, because all of these are industrial cases where you're trying to cure problems.
But if you're hungry, you need to go to survival mode. If you're a microbe,
and
the survival mode can be the store form. You get all sorts of, let's say, the rhizoctonia and agar blocks and things. Really interesting in terms of how you can induce reproduction. So again, that was part of my concept of that study was, again, industrialists might be interested in terms of.
That's why my PhD would have got done, because
I think
I told the nutrient where with high nutrient realised actually I shouldn't have been doing that.
You mentioned that they could reproduce vegetatively. So does that mean plowing's a rubbish idea or a good idea?
Okay. So ploughing is in general a rubbish idea because most of the fungi in the soil system are beneficial, and every time you plough or disturb the soil, you're ripping those mycelia up. They can regenerate, but in order to regenerate, they're using energy to do that regeneration rather than energy to do the cool stuff that you want to do to make your plants grow better, etc., etc. And if you continually do it, you will run them down in those terms. The other thing that can happen is a bit like couch grass or whatever else. So remember Rhizoctonia solani, the black skirt and stem fungus? This is a curious one. It's a great lab rat because it doesn't sporulate. It's never been known to sporulate in the history of science, so it's really good for you to have these spores everywhere. But of course, that then can reproduce by fragmentation. So if you fragment the Rhizoctonia mycelium, it's like couch grass, you're just spreading propagules everywhere. And they're adapted to regenerating from that, and you'll get a more pervasive disease. Ploughing in general, discourage it enormously.
Yeah.
Carl, is it right that mycorrhizae can work at pressurized PS pressure of 1100 psi downwards into the soil, so it's not anaerobic?
So I don't know about 1100 psi, but certainly, as I said at the beginning, fungal mycelia are hydrostatic systems. They're bags of pressurized liquid, and so forces. Any physicists in the audience, forces at these microscales are rather different to those at the macro scale. Okay, so 1100 psi, if you're 3 micrometers in diameter, is not such a big deal. The other thing is that we know that so the fungi pressure is 2 examples. One is the classic one is mildews. Is mildews. When mildew grows on the leaf surface, it needs to penetrate the leaf, and what the mildew fungus does is it forms a little pad that's called aprosorium. And it then has an infection peg that comes out of that and punches its way into the leaf. And basically, the little pad glues onto the leaf, and the peg pushes down. And the forces involved here, I think that's probably where the 1100 psi is coming from. I'm less convinced about mycorrhizae for different reasons. But so the mildews are real crackers on that front. And the other one is actually this little beauty, the Lloyd's wig fungus again. Massive aggregation of hyphae, you can see there and so forth, and there are many recorded instances of these fungi, because they often come in clusters as well. They're the ones that can crack asphalt and push paving slabs up. So the pressures involved there, again, physicists, you can calculate how many newtons you need to lift a paving slab. So that's well documented as well. And it's because, again, you've got the aggregation of all these items and stuff, a big pressurized system, and that can then scale and do the heavy lifting.
Any more questions? Yeah.
Until recently, I'd always assumed that fungi was something that occurred in terrestrial environments. But I read in an article recently that they're quite common in marine environments as well.
They are ubiquitous. So marine fungi is a whole subset of mycology. I'm a terrestrial ecologist, so I'm giving you all these. But
are they separate species?
Yes, they're separate species. Yeah, they're occupying their different ecological niches. They are still largely mycelial forms. Not sure if there's yeast in marine systems. Yeah. So mycelial form, and again, they'll be free living, they'll be parasitic, they're fish parasite. Well, we all know, we've all seen goldfish with fungi hanging off, haven't we? What's the name of that? Yeah. And so on and so forth. There's also a fascinating subset of fungi called the aquatic hyphomycetes, and these guys are adapted to living in frothy water, freshwater foam. So next time you see some foam on the streams around lots of them, those foam films will be heavily colonized by these hyphomycete fungi. They have specially shaped spores that are adapted to the surface tension stuff that's going on there, etc., etc. And they're involved in decomposing litter in freshwater streams and things. A whole, very, very diverse. So yeah, truly ubiquitous. Less so, less sexy. And again, look them up, and there are reviews around this. Yeah.
And those perhaps not pure cultures either. By the time we've got them, we've got a whole load of other organisms.
Yeah, that's right. So again, everybody please remember that microbiologists tend to be preoccupied with individual species, but actually it's all about community context and community interactions, and that's again one of my overriding research interests. That's the story from the morning.
Just to address your question about marine fungi, I've just finished coordinating the report in regulations that include marine fungi in relation to human health. And also a role in communicating fungal resistance genes and the fungal resistance. And fungal diseases in humans are becoming, as Karl mentioned, quite a lot of them. Things like black fungi, the slime, which is very prevalent in India, and becoming worldwide concern, particularly with growing resistance to antifungal agents. So it's a big potential human health and public health problems. Any more questions?
You mentioned that the evidence for nutrient transfer that the boy wrote is not actually very clear. What about sort of communication, information transfer in terms of plant affairs?
Yeah, so there's a few high profile experiments that have shown this around the signalling. There is some discussion and debate around the potential of volatiles in that. So it could well be volatile signaling rather than natural transduction via the mycelium networks. So it's back to these things are not straightforward. It's getting the stringency of the conditions such that they're really, really locked down. And of course, the other thing is that if you set up a hyper stringent circumstance, that will never happen in nature. And if there's a cheaper way to solve this issue, whatever else, then that will go on. But watch this space, remember when David Attenborough was told the continental drift was complete nonsense by his geology lecturer at Cambridge? So just because Professor Ritz says it's nonsense doesn't mean that actually the Woodbine Web is zinging along there. It's just back to this business again. This is important in this sort of forum where we have to get this balance between rigorous hardcore science and the chatter, and especially the attractive chatter that goes on there. So always, review the evidence with care.
Any more questions?
Questions, thank you.
Yeah. Okay. also interest and questions. Well, that's a very important part of this process in helping public understanding of science. Thank you very much and stay forward.